Hi, today we'll cover diffusion, but before we get started, we're gonna explain a little bit about the introduction, referring to cellular membranes and cells in general. As we know, cells are mostly made up of water. Maybe about 75 and 80% of all cells have that amount of water. Um, Cellular membranes are also better known as biomembranes, which are a really essential component of all living organisms. They are really important for regulation and trafficking of ions and molecules in and out of the cells. Um, because they are really thick membranes and awesomely plasma dynamic, and their structure allow it to be as permeable selective. Um, Membranes carry out many vital cellular activities, including cell compartmentalization, protein storing, ensuring the cytoskeleton, and production of energy such ATP and NADPH, cell signaling, nuclear division, and addition of cells to each other to the extracellular matrix. Um, as we also know, phospholipids, protein, and carbohydrates are the three main components within a cell, and they form the fluid mosaic model. These phospholipids are antipathic, but what do we mean by that? Antipathic means that they have hydrophobic and hydrophilic regions, nonpolar and polar regions. Well, let's get started. Diffusion. Um, Diffusion is the movement of cells. It's the movement of solute from a region of high concentration to one of lower concentration. The rate of diffusion is affected by such factors as temperature and the size of the solute molecules. We have two different kind of transfers. In this case, is passive transfer and active transfer. But whatever we mean by that is that substances can also move across the membrane via facilitate diffusion, which is part of the passive transfer, where a transport protein serves a pathway to facilitate movement of substance. Energy is not needed for passive transport to undergo conformational changes. Conversely, active transport uses a source of energy such as ATP hydrolysis to move concentrations from areas of low to high or against a concentration gradient within the cell. Our experiment today, it's to use dialysis tubings to recreate the cell membrane, which are selectively permeable to solutes based on size, charge, and polarity. In this experiment, as we see, that's a dialing tube. We will use about 25 millimeters of starch and put it in the 25 centimeters length of dialing tubes. After that, we're gonna place it on a bit of after we submerge the dialysis back into the beaker of water, we're gonna let it run for 60 minutes. During that period, we're gonna have eight tubes, and we're gonna numerate it 1 to 8. We're gonna pour 20 millimeters from the beaker into a 25 millimeters graduated cylinder. We're gonna pour 5 millimeters of each of the solute into each of the test tubes from 1 to 4, and then 5 milliliters of the solution into test 5 and 8. Today we're gonna test a presence of starch, of sulfide ions, chlorine ions, and proteins. The test tube solution will turn a dark blue-black color as a positive result after adding drops of Lugol solution. To test sulfide ions, a white precipitate will form after adding 2% PLA solution if there is a presence of sulfide ion. To test chloride ion, if a chloride ion are present, a milky white precipitate will form after adding several drops of silver nitrate. And to test protein, a positive result will show a presence of protein if the solution turns light lavender after adding drops of beetroot. The effect of temperature on diffusion. The goal of this experiment is to determine the direction of solute's movement based on the size and presence of the concentration. In conclusion, what we learned today about diffusion. The dialysis tubing is a selectively permeable material that provides a means to demonstrate the movement of substances through cellular membranes. Today we demonstrate that the direction of solute movement are based on size and the presence of a concentration gradient.